They need a special transport system to decrease the distance of diffusion between the nutrients and the transport system in the plant is the vascular bundle. The vascular bundle is made of xylem vessels and phloem seed tubes or phloem tubes. So this is the vascular bundle in the plants. The xylem has two main roles. The xylem is to support the plant, and it's for the transport of water and minerals from the roots to the leaves. That's why the movement in the xylem vessels is always upwards, from the roots to the leaves. Because the source of the water and the minerals is in the soil, and they are being transported to the leaves. But the phloem is for the transportation of sucrose and amino acids. And the translocation of the sucrose and the amino acid is from source to sink. So the source is where the food is, where it's going to be made. And the sink is where it's going to be used or stored. So the source and the sink will differ according to the age of the plant. In an adult plant, the photosynthesizing leaves are the source. This is where food is made. The sink can be the roots, the tubers, can be the growing tips, the fruits, they can be sinks. So the phloem, the movement can be downwards if the sink is below the source, or it can be upwards if the sink is above the source. If we have a seedling, which is a young plant, just germinated, that's the radical and that's the plumule, and those are the baby leaves. In this case, the source is the seed itself, the cotyledon. And these young leaves will be acting as sinks because they are not large enough to make photosynthesis for the whole plant, which is an adult plant. A vascular bundle is extending throughout the whole of the plant, from the roots up the stem till the leaves. Okay. If we have a look at the xylem vessels and how they are adapted to carry out their function, the first adaptation of the xylem vessels is to form a continuous column. I need a vessel extending through all the plant, from the roots till the leaves. So these cells start to arrange on top of each other. They are stacked end to end to make a vessel. And then these cells start to make the lignin, and the lignin is deposited in the cell walls. So lignin starts to thicken the cell walls, forming the lignified walls. And this lignin is for support. When the lignin is deposited, there are tiny areas in the walls with no lignin, and these forms the pits through which the water can move in and out. It's for the lateral movement of water through these xylem vessels. The lignin is waterproof. It's impermeable. It will prevent any nutrients from entering the vessel. And that's why the contents of these cells starts to die. And they start to form a hollow vessel with no cell contents. They are not large enough to supply the cells with all the nutrients they need. to maintain their vitality. But at the end, all these cells die. And all the contents, even the end walls, get broken down. And they start to form these hollow vessels. And this is very important for the easy transport of water and minerals. When they are hollow with no cell contents and no end walls, the water with the dissolved minerals can move easily up the xylem without any resistance. The cell walls of these cells become thickened with lignin. Lignin is a lipid substance. It's going to be made by the cells and they deposit it in the cell wall. So the cell wall becomes strong. Okay? The phloem sieve tube is for the translocation of sucrose and amino acids. The translocation of sucrose and amino acid is an active process, which means that these tubes must be living, must be a living tissue. It cannot be dead like the xylem vessels. The movement in the xylem vessels is a passive process. It doesn't require energy. But the movement in the phloem requires energy. So these tissues must be living. Again, it's forming a continuous tube throughout the plant. So the cells that are going to form this tube are arranged end to end on top of each other. Then all the cell organelles get to one side of the cell. All the organelles with the vacuole and the nucleus and the RER, all the different organelles and the mitochondria come to one side of the cells. And this will be known as the companion cell, which is carrying all the cell organelles. And it leaves the rest of the cell nearly empty, just a very thin layer of cytoplasm next to the walls, but the cell itself is empty. And the end walls start to become pitted. They start to form a sieve plate. This makes it easier for the sucrose to move from one to the other. The sucrose just passes through these pores, through these pits. Instead of crossing the cell membrane from cell to cell, it just passes through these pores, so it's a faster and easier movement. Okay? And at the same time, all the cell organelles are at the side in the companion cell, so they are leaving the center of the tube nearly empty so that the sucrose can move easily without any resistance. Okay? Transverse section in the root. So that's the root, and we're going to take a transverse section. We will find the outer layer of the root, which is known as the root epidermis. That's the outer layer of cells that starts to differentiate and form root hair cells. So cells of the root epidermis can differentiate and make the cytoplasmic extensions and form the root hair cells. Epidermis is the outer layer. The leaf عندها upper epidermis and lower epidermis. The roots have an epidermis, the stem has an epidermis. That's the outer layer of cells. In the center of the root is the vascular bundle. So the vascular bundle will be central here. And the xylem will show a cross arrangement in this center. Always the xylem, when it is drawn, it will have thickened walls. The walls will be in balls because it's thickened with lignin. So that's the xylem in the center, in the arrangement of a cross. And the phloem is arranged in between the xylem. So that's the phloem. This is the phloem, and this is the xylem. Okay? The cells from the outer epidermis till the vascular bundle, these are all undifferentiated plant cells known as the root cortex. Cells of the root cortex. Undifferentiated plant cells means they are plant cells with the usual structure of a plant cell, which means it has a cellulose cell wall, it has a large vacuole, it has a nucleus, the RER, but it doesn't have any specific function. So it doesn't have a specific structure. Not like the palisade cell, for example, whose function is to carry out photosynthesis, so it has a lot of chloroplasts. Or like the root hair cell, whose function is to absorb water and minerals from the soil, so it has a cytoplasmic extension. These cells, they are just plant cells without a specific function, so they don't have a specific structure. Okay? In the stem, the vascular bundles will be arranged all around the periphery. So each one of these structures is a vascular bundle. And they are arranged peripheral like this to give more support. So each one of those is a vascular bundle. No, that's the root, that's the stem. And in these vascular bundles, the phloem is towards the outside, is towards the stem epidermis. So all the outside is the phloem, and the xylem is towards the inside. Below the phloem is the xylem. And all these areas are filled with cells of the stem cortex. Again, in the leaf, the vascular bundle shows the xylem towards the upper epidermis and the phloem towards the lower epidermis. So that's the arrangement in the leaf. Transpiration is the evaporation of water from the surface of the cells and the diffusion of water vapor from the opened stomata of the leaves. So transpiration is not only the diffusion of the water vapor out of the stomata, it's also the evaporation of water from the surface of the cells. Any cell exposed to air, the water inside the cell can start to evaporate. So we can have evaporation of water from the upper epidermis or from the lower epidermis. Or mainly from the spongy cells, because the spongy cells are surrounded by large air spaces. So the water on the surface of the spongy cells keep on evaporating. 98% of the absorbed water is lost in transpiration. 2%. Only 2% of the water absorbed remain inside the cells. Only 2% remain to be used in photosynthesis, to be used for hydrolysis as a transport medium, to dissolve different substances, for turgidity and support. Only 2%. 98% are lost. And that's really very important, because without the 98% that's lost, the 2% needed by the cells will not be available. Because when water is lost, this is going to create the water potential gradient, which will pull the water from the soil. It will not. The water will not move. There will be no water movement. If it doesn't use this water, the water movement in the plant will stop. And the mineral movements will stop. Don't ever forget that the minerals are dissolved in the water and they are moving up the xylem with the water. So if the water doesn't rise to the leaf, even the minerals will not rise to the leaves. Yes. Okay, let's see how this is going to happen. So the source of the water is in the soil. That's the high water potential found in the soil. And it enters the plant through the root hair cells by osmosis down water potential gradient. And then this water starts to cross the cells of the root cortex till it reaches the xylem in the center, till it reaches the root xylem. The water starts to move through the cells of the root cortex from cell to cell. Yes. If you remember again, to start to recall again the transverse section in the root, that was the root epidermis with the root hair cells, and that's the vascular bundle with the xylem here in the center, and these are the cells of the root cortex. So the water is entering from the soil through the root hair cell. That it has to cross all these cells till it enters the xylem. And of course, the movement of water is always by osmosis down water potential gradient. So the water is not entering the flowing phase, and we are concentrating on the xylem because this is what's going to transport it. So it's going to move from cell to cell by osmosis, or through the cell walls and the intercellular spaces till it enters the xylem through the pits in the walls. And then the water starts to rise. So the water rises up the stem xylem till it reaches the leaf. The water rises up the stem by three forces. And the main force pulling the water upwards is due to what's happening in the leaves. Because the force that happens here, which is the osmotic root pressure, this is just enough to make the water rise just a little bit. It's because the water is entering with a very large amount in a tiny tube, so the pressure is very high, it starts to push the water upwards. So this is going to push the water upwards, but just for a very short distance. But if you have a very tall tree, this pressure is not enough to push the water up to the leaves. You need something stronger happening in the leaves to start to suck this water upwards. You need a force to start to suck the water upwards. Okay? So what's happening in the leaf? In the leaf we have the mesophyll cells, the palisade mesophyll, and the spongy mesophyll surrounding the vascular bundle, surrounding the xylem. So this is the spongy mesophyll, and these are the palisade mesophyll cells. And we also have the guard cells and the stump-tubular epidermis. And the spongy cells are surrounded with very large air spaces. So what's happening in the leaf is that the palisade cell is taking water from the xylem because it's making photosynthesis, and the spongy cells also are absorbing water from the xylem by osmosis, so these cells are filled with water. The main concern will be the spongy cells. Why? Because they are surrounded with the air. They are surrounded with the air spaces. But because these spongy cells are surrounded with large air spaces, the water will start to evaporate. You know, anything, if your hands are wet and you keep them in the air, what's going to happen to your hands? They will dry. Why? Because the water will evaporate. So that's exactly what happens here. The spongy cells are wet because they have a lot of water, but they are surrounded with air. So the air will cause the water to evaporate, and it starts to form water vapor in these air spaces. And water vapor starts to diffuse out of the open stomata. So the spongy cells keep on drawing even more water. So whenever the spongy cell is drawing water from the xylem, this water evaporates and diffuses out, so they start to take in more water. This absorption of water from the xylem, this is the suction force. This is known as the transpiration pull. So there is transpiration happening here. The spongy cells are losing water, but due to this loss of water, they start to take in water from the xylem. So they start to suck this water, they start to pull it. Okay? And because the water molecules tend to stick together, there are strong cohesive forces between the water molecules. The water molecules stick together and they stick to the walls of the xylem. They make strong forces with the xylem walls. These are the adhesive forces. And they stick together by strong forces. These are the adhesive forces. So because they are stuck together strongly, when one water molecule is pulled, the whole column is going to be pulled. So when the spongy cells start to pull on one water molecule to absorb it by osmosis here, it actually pulls all the water of the xylem, of the whole stem xylem. Yeah, and now it's losing water. The water in the spongy cells are evaporating and diffusing out of the stomata. So they take in more water from the xylem. So they start this transpiration. So the transpiration pull happening at the mesophyll layer, between the mesophyll layer and the xylem. This creates the suction force, which pulls up the water due to the cohesive and adhesive forces. Cohesive forces are the forces between the water molecules together. Cohesion of water molecules, they stick together. Adhesive forces is the forces between the water molecules and the walls of the xylem. They stick to the walls of the xylem. And this is important, so when they are pulled, they do not break. They can rise all as one column. Okay? Understood?